Today, Aarash Bordbar is going to give the lecture here. He graduated last year with his PhD in bioengineering. He is now involved in entrepreneurship, for those of you interested in starting companies. Towards the end of his thesis, he dug out the entire literature on the copra field, starting from 2000 roughly, and put together a review that came out in Nature Genetics reviews, I guess, early last year. It was a little over a year old. And as he analyzed the literature, he discovered that somewhere around 2010-11, there was a big shift in this field towards real predictive biology. So in other words, how do the methods taught in this class lead to prediction in biology? And prediction, of course, is a valuable thing. So he picked out 10, 12 key papers that are in this review article, and he's going to go through the paper with us here today. It's just a great way to set the stage for the last part of this class, where we go through some of the applications of copra methods. So Aarash, welcome back.
Okay, so today I'll be talking about this review that I put together in late 2013. And it's called Constraint-based Models Predict Metabolic and Associated Cellular Functions. And this is the citation for that review if you're interested in looking at it. I'm sure it's available on the class website or something like that. So let's just begin with the title of the review. And the main word that comes to my mind that isn't fully defined here is the word predict. So what is prediction? So I went to the dictionary and I looked up what they define as predict. And it's to declare or indicate in advance, especially foretell on the basis of observation, experience, or scientific reason. Some famous people have had some quotes on prediction. So Niels Bohr said prediction is very difficult, especially if it's about the future, which is a little interesting, I think. And Howard Stevenson, who used to be a faculty at Harvard Business School, he said prediction is at least two things, important and hard. So in biology, we strive to predict things ahead of time, either through scientific reasoning, through the scientific method, through, in today's world now with like bioinformatics and systems biology, we try to predict on a computer, and then we try to look at these phenomena experimentally. And biology is changing as a field, as I'm sure you're aware of, and that's why you're getting it. Taking this class, which is related more to systems biology and bioinformatics. And biology is going from a very qualitative, data-poor field to a quantitative, data-rich field. And this is really being driven by technological innovations that allow us to measure various biomolecules both comprehensively and in the last decade very cheaply. And for example, here's the cost of sequencing a megabase pair over the last decade. And the price has been dropping astronomically. And this is compared to Moore's law, which is the change in price of and the amount of transistors you can fit on a chip. And so these technological innovations have really changed how we do biology. And the inflection point was basically in 2008, which is around when I started my PhD. And so let's just talk about the scientific method, or kind of the traditional way of how biology is done. And so you define a question, you gather information and resources, you form a hypothesis, you then test that hypothesis, you analyze data, you draw conclusions, and then you retest either yourself or other third parties. And this is how biology and most sciences have been done for hundreds of years. But in 2008, there was a controversial article in Wired magazine about how the scientific theory is now dead. And it was saying that because there's so much data available, there's all this big data, we don't need theories anymore. We can just do analytics on humongous data sets, and we'll be able to tease out the hypotheses straight from that. We don't even need to make a hypothesis. We don't need to build models. And this was written at a time when the Google Flu project was doing very well, which was trying to predict how the flu spread based on people's search terms in Google. And it was doing really well in 2008. And by 2014, the Google Food Project ended up being a huge fiasco, and it made a lot of really bad predictions. And so there was another editorial at the time in the Financial Times around that time, around when the Google Food Project failed, that basically said we might need to temper our feelings on just going with statistical approaches and big data, that we need to actually try to still tie it back to mechanisms. And in biology, we're able to measure various biomolecules, as I alluded to. We're able to measure things that happen, or the DNA itself through DNA sequencing or SNP genotyping. We can look at how proteins interact with the DNA. We can look at how the transcriptome is composed, how RNA is transcribed either through RNA-seq or microarray technologies. We can look at ribosomes, we can look at proteins, we can look at even metabolites. And so we can measure all these different type of data types now. They're getting cheaper to do, they're becoming more comprehensive, there's less noise in the data. And data generation is always getting better, but it's starting to seem that the real bottleneck is no longer data generation, but it's data analysis. How do you take 5, 6 levels of data, integrate it together, and make meaning of it, especially if you want to do it from a mechanistic standpoint? And Dr. Paulson and I put together a review which this lecture is on, and in it has this table, which is different methods for doing biological data analysis. And don't worry about the text here. There's a lot, you probably can't read it from there. But what's important to notice is it goes from statistical approaches at the bottom to kind of more logical rules modeling, to constraint-based modeling, and then all the way up to stochastic kinetic modeling. And the important thing to remember here is that as you go up this list, you increase in mechanistic complexity of what you're modeling, but it's always at a trade-off of how much coverage you have of a biological process or what biological processes you can even model. So it's important to remember that trade-off when deciding on which type of modeling approach to use. And the main areas that are discussed in this review are related to constraint-based modeling, and there's some stuff related to kinetic modeling as well. So the constraint-based modeling field has been growing. There's a whole host of different methods that are now available. Nathan Lewis did a review on this a few years ago, and at the time there was about 100 different methods for analyzing, interpreting, or building these type of models. And when we put together the review, we went to the literature. We amassed about 20,000 papers from the early 80s, and we kind of try to track when did this field start and how has it been growing. And so the first prediction was in the mid-80s, and since then it's been growing exponentially in terms of the number of publications related to how the constraint-based model is used to make a biological prediction, and then being subsequently validated either through prospective experimentation or retrospective, data analysis or literature writing. And so the field has gone through basically 4 stages. Stage 0, I guess, is the pre-genome era, before genome sequences were used to build these models, and it was just putting together maybe just glycolysis and pentose phosphate pathway together. Then moved on to the genome scale level. That's where there's been a lot of conceptual development, building different methods like pathway analysis methods, knockout techniques, etc. In stage 2, omic data became easy to generate and relatively cheap, so they started to be integrated with the models. And then what this review is concerned on is what's happened since about 2009, 2010, where people are so comfortable with these methods and it's becoming accepted that experimentalists will actually test the predictions that are being made, and they are often able to find some pretty extraordinary things that are non-intuitive. And this is the applications for these methods are in a lot of different fields: metabolic engineering, infectious disease, environmental sciences, biomedicine, and synthetic biology. And there's over 100 organisms that have been modeled. The 3 main ones, which account for I think more than 50% of the papers, are E. coli, yeast, and human metabolism. And as of I think early 2014, there was about 650 publications. And if you would like, you can go to this website, which is listed at the bottom, and it has all these papers cataloged there. with metadata in terms of what the application was, what method they used, what the link is, what organism, authors, etc. So it's a great way to kind of read up on what's already happened in the field if you're trying to decide on new projects to do. And I'd also like to mention that there's been, as I said, there's a lot of applications now in human metabolism, and that's continuing to grow. I guess this is the one area that was the smallest when the review was written, but it's probably the biggest area that's going to move forward, is to look at human metabolism. So let's just start with a quick history of the field. And the idea of using constraint-based modeling goes all the way back to the idea of yield. And there's this very famous paper from the early 60s on how the dry weight of an organism produced was proportional to the weight of the energy source. So the idea of the amount of substrate you give something will result in the size of the organism or the amount of cells that you get at the end. And this was a pretty new concept, I think, and it was shown here quantitatively. And this is a very important idea that kind of goes back to why these models are used, because the traditional calculation that a constraint-based model does is a yield analysis. And there's basically two methods that kind of made up what became constraint-based. And one is optimization, and the earliest I could find in the literature about that was this paper by a Professor Watson out in England, I believe, in 1984. And he had written an Apple II program for his students so he could teach them glycolysis and pentose phosphate pathways. So he wanted to show them, instead of writing out the equations of the stoichiometry and figuring out that one glucose gives you 2-pyruvate, for example, you could do it through a computer program. And this is the original model that was used. Another main concept for the field was looking at convex vectors. So this was started by Bruce Clark back in the 80s, and then this figure, showing an example of it, is from a paper in PNAS. And what is important here is, as you guys have already learned, there's a solution space to these models, and the extremes of this space, or the convex vectors on the edges, actually make up biochemical pathways, which is a very intriguing idea that this math concept is linked to something that is related to biochemistry. So as I mentioned, the first prediction or use of these models for prospective prediction was in the mid-80s, 1986. And basically they built a small model of an adipocyte and showed that lactate formation is not stoichiometrically required for fat synthesis. So if you optimize for some sort of fatty acid production by the cell, it doesn't need to actually produce lactate. So they're not stoichiometrically linked. Obviously the cell still produces lactate, so there's other biochemical processes occurring that cause that, but just from a stoichiometric standpoint and from a yield perspective, you don't actually need to generate lactate. And this kind of leads to the need for an objective function that has often been used in constraint-based modeling. And there's been a lot of computational experimental work to try to figure out what do microbes strive for, right? So just the idea of having an objective function almost implicitly requires the modeler's thought process of what do we think the organism is doing or trying to achieve evolutionarily. So the initial work that also got people really interested in this field was looking at is the cell striving for growth. And there's been work done that showed basically the cells, when you would optimize the model for growth, you would get some sort of yield that the cell would not be able to do when you grow it in the lab. But if you adaptively evolve those cells, they actually move towards that objective function value. So when the cells become optimal, as they try to adapt for maximum growth, they will actually reach this computation of predicted maximum growth. A paper a few years later showed that it might not be just trying to grow, but it's also for producing energy. And they use those convex vectors, those extreme pathways, as I mentioned earlier. And what they showed was that it's actually a trade-off between growth and energy. And they had a third term, which was almost like a fudge factor. They didn't know what this third term could potentially be, and they said there was some slack in the model, but it definitely, two of the important points is growth and energy. A few years later, using C13 analysis, Uwe Sauer's group showed that in fact there's potentially many different types of objectives. And they went through the literature, they gathered up different objectives that people had used for the models, and then they compared it to different scenarios of the cells growing, either like in batch or in chemostat or in different conditions. And they saw that for different conditions, a different objective function was suitable in terms of how close the prediction was to the experimental fluxes. And in this paper, they kind of just said, maybe we don't know. Maybe there isn't a real specific objective. But in fact, a few years later, the same group showed, and this kind of relates back to that second paper I mentioned, that there's actually three basically trade-offs that the cell is optimizing for. So the previous paper I mentioned, remember, had two. It was energy and growth of the cells. And so it turns out that it ends up being those two factors, the biomass yield and the ATP yield, but it's also the sum of the fluxes, or how the cell is trying to make the best use of its proteome and minimize, overusing of different of proteins and enzymes, and keeping the fluxes overall down. So the second paper I want to talk about is related to enzyme specificity. So the traditional biochemical textbook says that enzymes are powerful and highly specific catalysts. And there is literature and theory is that ancestral enzymes were inefficient and promiscuous, that they weren't weren't evolved to the capacity they are now. But there are some modern enzymes that are highly specific and efficient. So your textbook says that it's all enzymes are highly specific and efficient. In reality, only some are, and a lot are inefficient and promiscuous. Right? So why? Why did some enzymes become highly specific while others remained promiscuous? Ho Jung Nam and Nathan Lewis put together a study where they were trying to figure out what were the reasons from a network perspective that make these enzymes become specific while others not. And basically they were trying to figure out what is the metabolic properties that influence enzyme evolution. And the idea was that there was potentially pinch points, or stress points in the network that forced these enzymes to become specific for some sort of evolutionary advantage. And so they looked at a few different characteristics of both the generalist and promiscuous enzymes in E. coli. The first is that they saw that in general, the specialist enzymes that are in E. coli carry a higher flux. So they've become specific to reduce enzyme mass and to kind of get rid of, not have waste. The second portion that they looked at was that these selective enzymes end up being more essential than the promiscuous ones. And they show this both in terms of the KO collection in vivo, what are essential, as well as using modeling techniques. And third, they saw that specific enzymes were more regulated, both transcriptionally and post-transcriptionally. That when you shifted the cells through different substrates or growth environments, the ones that changed the most and had to be regulated were related to the specific enzymes. So the second portion of the review looks at characterizing interaction networks. So there's other types of biomolecular networks, genetic interaction networks, transcription regulatory networks, and these studies try to use the metabolic model to kind of give you new principles that you could learn from when comparing the metabolic network with those other types of networks. So the first is a genetic interaction network. So Zapanos in 2011, they put together a project where they calculated what are the in silico genetic interactions between different genes in yeast. So it's finding whether a double knockout has synergy or antagonism versus the single knockouts. And what they found first was that the calculations, even though they weren't that accurate compared to actual genetic interactions, they still kind of had the same global properties. And the main one being that the single whatever the gene mutation's fitness was in the single knockout, it was inversely related to the degree of the epistasis, which is kind of counterintuitive. And it's because that they proposed that it was related that those single genes were occurring in biomass precursor pathways. So when those were so perturbed that there would be less epistasis. They then also used the network and compared it to the metabolic model predictions, and they basically tried to figure out a machine learning way to fix the discrepancies between the genetic interaction network, which is experimental data, versus the metabolic model's predictions. And so they came up with a machine learning approach that just either changed the directions of reactions in terms of allowing it to be reversible or not, or just completely removing reactions to try to best fit these genetic interactions. And a lot of the prediction error occurred because of a pathway that said that you could make NAD from aspartate. So that is included in the model because it was in the biochemical textbooks. It's in E.coli, and most metabolic network, or not metabolic network, but most people in metabolism typically think that the same metabolic components are in essentially every cell, and that just happened not to be the case. So that pathway was included from old literature on biochemistry, but the actual genetic interaction data was saying that these two other pathways were having a synthetic lethal with each other. So the model obviously would not predict a synthetic lethal because it had a third pathway that it could produce NAD from. So they went in and experimentally verified that in fact that pathway is not there, and fixed the biochemistry. The second paper related to looking at interaction networks was looking at transcription regulatory networks. And here, this group calculated different pathways for these networks using a similar approach to extreme pathways. And what they did is they then clustered gene expression data from the many microbes database, which has thousands of expression profiles. And what they saw is that the ways the pathways were built by the network, they're very co-regulated, which is a good result. You'd expect that a pathway would be co-regulated if it's a particular function for the cell. But what they found that was really interesting was that some pathways were nearly 100% co-regulated, while other pathways were not co-regulated at all. And they kind of wanted to figure out why that was. And it turns out that it has a lot to do with protein cost. And the amount, basically by protein cost I mean the size of the actual protein that's in the pathway. What they found is if the pathway consisted of proteins that were all very hard for the cell to produce in terms of the size of the protein, then it would be co-regulated a lot. Like all the metabolic enzymes would be transcriptionally regulated because the cell just doesn't want to expend so much energy. But if the pathway was filled mainly with proteins that were very small and didn't have much cost to the cell, in fact, the only regions that were ever transcriptionally regulated at all were the first enzyme and the last enzyme. So that's kind of an interesting interpretation of why the transcription regulatory network is how it is, and it was guided by how you were able to do the metabolic pathway analysis. Another important use of these models has been kind of expanding what are metabolic knowledges, and I alluded to that on the study with the yeast genetic interactions, right? So if you have experimental data, you have computational results, and they don't match, and there's a discrepancy, then there has to be some reason why that's occurring, and it's typically that the model is based on whatever literature that already exists. There must be a gap in that knowledge. And so there's various different types of modeling gaps. The perfect scenario is that you have the gene you know which protein it becomes, and you know what reaction it's catalyzing. That's the best scenario. You might have cases where you don't know what the genetic makeup is for that protein, but you know the protein's there. You might have where you know there's a gene, and you know in E. coli there's a lot of Y genes, but you have no idea what their function is. You might have reactions in the model that you know have to be there for the model to function, but there's actually no annotation whatsoever of, you know, why that's in the cell. And then there's also, you know, network gaps that occur that you know lead to dead ends or cause the network not to fully function because you're missing particular reactions in a pathway. And my favorite paper actually in this entire study is this one, the Nagagashi paper, where this group wanted to test out and determine if there are any gaps in E. coli central metabolism, which is pretty interesting, I think, because E. coli central metabolism is probably the most well-studied metabolic network there is, and they were able to find that we still don't even know some things there, which is, I think, incredible. So what they did is they came up with this new technology to do double knockouts really quickly, and so they did single knockouts, double knockouts, and even triple knockouts of central carbon E. coli genes. And they looked at the epistasis there, you know, what the synergy was occurring between multiple knockouts. And at the same time, they wanted to see what the literature said. So their version of what the literature says is basically, what does a flux balance model tell you for these same predictions? And what they found was that, you know, in general, the flux balance model was actually very good at predicting all three levels of knockouts. But in terms of TAL, which is related to the pentose phosphate pathway, almost all the predictions there on the double knockouts were wrong. And so the model in general is pretty good. They had some errors, but most of the errors they could, they knew what the reason was just based on the assumptions of the modeling approach. But for some reason, predictions related to the gene TAL were usually just wrong. And so first they looked at the knockouts, and they saw that the gene expression levels were not different. And so the error was that the cells would still grow, but the model was predicting that the cells wouldn't grow. So they took those cells that were still growing, they looked at the gene expression. The gene expression had basically not changed. So there's no way to look at the expression profile. And so then they started looking at the metabolite levels. And they identified a molecule that had never been known to occur in E. coli, which was, so when you create S7P in pentose phosphate pathway, it needs to be degraded by TAL. So the model, obviously, when you remove that reaction, it can't degrade it. But when they did the metabolomics on these mutants, they saw that there was a new molecule, S17P, which is a diphosphate version of that metabolite. And it turns out that they did extensive in vitro tests, and they saw that PFK was actually phosphorylating S7P to become S17P, and then the aldolase in glycolysis was then cleaving that to allow it to go back to glycolysis, which is essentially a new mechanism for how a cell is robust, that it's able to take a metabolite that's building up and use other machinery that is potentially thought to be very specific, but is in fact promiscuous in the sense that PFK can then do this additional function, which I think is very interesting. Another study in this category was related to human metabolism. The human metabolic network had the molecule gluconate in it. It's known to be in diets, but the model and literature just had no way of, there was no analysis for how gluconate could be broken down to other elements. And so what Otter did is he looked at what potential pathways or reactions you would need to connect gluconate to the rest of the metabolic network, and he hypothesized that it was related to become 6-phosphogluconate. And then he did mass spec analysis and found that it was related to this gene region C9orf103, which was in fact the gene that was doing this phosphogluconate activity, phosphoglucomutase activity. Another category is looking at designing metabolic phenotypes, or coming up with genetic engineering strategies using a rational modeling approach. This has mainly been applied to metabolic engineering, and the idea for this algorithm OPNOC and many other algorithms similar to it, and is one of the first main algorithms that was used in this field for predictive biology, is that if we look at the solution space here of looking at the growth rate and the product secretion, the cell will typically, as you grow it, it'll evolve to keep on growing faster and faster, right? And so if you even have a strain that is producing something, it could potentially just go back to that wild type condition where it's not producing it and it's just growing faster. So this algorithm basically gives you a list of knockouts that you should do to the cells such that the fastest it grows, it will still be producing that product. So this will allow you, one, to select for those cells just by their growth, and also the cells will never lose their ability to secrete the product. So you see there's kind of this dotted envelope where as it gets faster and faster, it's still producing that metabolite. And so this was reduced to practice in a paper in Nature Chem Bio, where they were trying to produce the chemical BDO, which is a commodity chemical, and they first added the pathway into the cells, but it wasn't really producing it. The enzymes required for it are not found really in any organism, so they had put together enzymes from different organisms to put this heterologous pathway in there. And after running OPNOC, what it was doing is it was removing the fermentative pathways of the organism, so it could no longer produce NAD or kind of recycle its redox. And then the only way the cell could potentially recycle its redox is to produce BDO, because that was the only way to get 2 NAD out, to kind of recycle for NADH. And then this was then scaled up and was moved to levels that were of commercial importance, which I think is the first in the literature to do that. There's a lot of other papers that do metabolic engineering strategies, but have never pushed it to the level where it could actually be commercially viable. And I think this is the plant that they're actually producing the BDO in. The next category is related to discovering drug targets. As you know, these models can do knockouts, so that's a very easy, or it's amenable then to try to look for particular drug knockouts. And this is pretty important, and as you can see here, there has been a drought in the pipeline, as they say, in pharmaceuticals being approved every year, and the cost of producing a drug has been going up considerably. You used to, for $1 billion, you could almost make 50 drugs in the 1950s, and this has been scaled for inflation, and now it costs nearly $2-3 billion to get one drug. And a lot of this has to do with just the regulatory environment, and this has actually changed, I guess, a little bit the last couple of years. And this past year, there was the highest number of approvals in the past decade. But it's still good to have new approaches to find targets and try to come up with new drug development strategies. So the first study was related to cancer metabolism, and Eitan Rupin's group had a pretty simple question, but it's tough to answer, is that a lot of cancers have metabolic oxytrophies, right? So the cancer cell has some sort of inability that a regular human cell does have. And so they wanted to exploit that. So they were looking, can we predict, if you take 2 drugs, can we predict a synergistic metabolic effect that would kill the cancer, but it would not affect healthy cells, ATP generation, and their growth? And they put together a great computational study, and they saw, they found, I think, 50 combinations, and I think 30 of them were already either current drugs or going through the regulatory process, so kind of validated the approach. So then they actually picked an example and looked at it experimentally, which they published in Nature in tandem papers. And this was focused on HLRCC, which is a kidney cancer. These cells don't have fumarate hydratase, which means that they have an incomplete TCA cycle. And they built 2 cell lines, ones that were fumarate hydratase deficient and a cell line that had the wild type fumarate hydratase in it. They built metabolic models for both, and they were trying to see could they use the fumarate hydratase as an octatrophy that you could then predict another target and make it a synthetic lethal, while it would not kill the normal cells. And so they did, gene deletion analysis with both models, the one with the fumarate hydrogenase and the one without, and they found one pathway that would preferentially kill the fumarate hydrogenase deficient cells, and then this pathway was related to heme biosynthesis and then its degradation to bilirubin, which is kind of a non-intuitive pathway. And what it comes down to here is actually, once again, redox. This pathway is needed for the cell that doesn't have a functioning TCA to be able to balance its redox. And the interesting thing is they looked at the gene expression data of these cells, and you can't tell that this pathway would be necessarily the most important, because I think this pathway ends up being the 13th highest differentially expressed pathway. So that would mean that you'd have to have done 12 experiments beforehand and potentially failed 12 times before you'd gotten to this pathway. Then they showed this both in the cell lines and in mouse models using small molecule inhibition of one of the enzymes as well as through RNAi screens. And here's the data for that. Another approach was looking for antibiotics, and they did a slightly different approach. Instead of looking at gene deletion, which is up here where you just iteratively remove the reaction associated with a gene, they did what's called a metabolite deletion. And so what you remove is the downstream products, the downstream reactions of a particular metabolite. So you're basically saying this metabolite doesn't exist, and then the downstream reactions can't occur. So they did this computationally. They found 193 different metabolite deletions that would kill this particular organism, Vibrio vulnificus, and then they filtered it down to 5 compounds. And the way they filtered it is basically they removed any compound that was structurally similar to compounds that occur in humans. So they were looking for very exotic compounds that have no similarity to human metabolites. And then to experimentally delete a metabolite, what you do, or what they showed works, is you just find a structural analog for that particular metabolite that you think is important and needs to be deleted. And if you overload the cells with that, it'll competitively bind to the proteins that are downstream and essentially block it. And so they showed this in vitro, and they found such a structural analog. They showed that it kills the cells, it kills it better than any known antibiotic, and they also showed that it does bind to the protein, the suggested protein that it was actually binding to. The last study in the drug target analysis, which actually could also have been in the genetic engineering category, designing phenotypes, is related to ROS production. So Jim Collins' lab has been championing the idea that reactive oxygen species are the main reason for how antibiotics work, and there's a lot of great work they've done that's shown that this is the case, and there's a debate in the literature, but what they wanted to show here is that you can actually genetically engineer an E. coli cell to overproduce reactive oxygen species naturally, and then it would be a lot easier to kill it with antibiotics because it's already in an almost antibiotic state already. So they took the E. coli model and they added they coupled it with the ROS generating reaction. So those are not explicitly accounted for in the original E. coli model. So they added tandem reactions for 133 known ROS generating reactions. And then they could, when they simulate the model, they can actually predict what the ROS generating effect is. And so they came up with genetic engineering strategies where if you knocked out a particular reaction, you wouldn't kill the cell, but you would increase its overall ROS production. And so they showed this experimentally, that they were able to predict and genetically engineer the cells to produce more internal ROS. And then they also showed that those strains, a lot of them, if you gave them antibiotics, you needed a lot less antibiotics to kill those cells because they were already in a susceptible state. The last category is related to coupling constraint-based modeling, which is a particular modeling formalism with other cellular processes that are often modeled with other modeling formalisms. And so this is about expanding beyond metabolism. And so I'll talk about 3 studies. The first is linking transcription and translational machinery to the model, so you can explicitly predict the transcription level and the proteome level at the same time as you're predicting fluxes. The second is linking probabilistic models of transcription regulatory networks with the metabolic model. And the last case is to integrate structural bioinformatics technologies with these models. And in those two cases, the idea is that you run your separate model, you figure out how that's going to affect an individual reaction, and then you integrate that with the network. And then you can see the systemic effect of it. So the first example is related to linking metabolism and expression. The expression and translational machinery is still formulated in a way in the constraint-based modeling formulation. And the idea here is typically you just have the reaction occurring, but here the cell has to synthesize the RNA, the protein, make the protein complex, bring it together to actually get the reaction to occur. And then you end up adding a lot of extra new reactions, right? So you're adding 50,000 reactions related to this machinery. And you have to couple these reactions to the model, which is an additional 30,000. So there's a lot of additional content you have to add. And the first paper to do this was by Josh Lerman, where they built a ME model, a metabolism expression model for Thermatoga maritima. And so the two results from this paper is one that you can compute the transcriptome and the proteome explicitly with the model. And also, they then used the approach for biological discovery. So they used it to see discrepancies between expression profiling data and the computational results, and they were able to find new biology. The second approach is related to linking metabolism and the transcription regulatory network. Here, Nathan Price's group, basically they took a large compendium of expression data, and they basically built a probabilistic model that if a particular transcription factor is on or off, then this is the percentage chance that the gene would be expressed or not. So then you could actually simulate a transcription factor knockout. And so what you would do is you would first calculate what would be the probability of each gene to be up or down regulated, and then they would take those probabilities and integrate it with the FVA bound, so the minimum and maximum bounds of the constraint-based model. They would adjust it by that probability, and then they could then do an FVA simulation. And they found that it was actually very accurate in predicting transcription factor knockout viability. The last study is related to adding protein structure info. So there's a whole field of structural bioinformatics. There's a lot of different methods like molecular docking, molecular dynamics. And what Roger Chang did was he wanted to do this at a genome-wide level for all the metabolic enzymes of E. coli, and then calculate a property for these, and then integrate them all into the metabolic network to get the physiological output of what this perturbation would potentially be. And so the perturbation that he was focused on was temperature. So he wanted to see how each individual protein was affected by the temperature, and then integrate this to see what the systemic effect is on the cell. So he built the structural proteome, as they call it, for this model, and then he used these structural bioinformatic techniques to predict the physiological function of the cell. So the first thing he predicted was the growth as a function of temperature. The cells grow near optimally at 37 degrees Celsius, and if you go outside this range by more than 5 degrees or so, the cells grow at slower rates. So he was able to computationally show that based on how the enzymes were changed and their catalytic activity was changed individually, and then how that integrates with the whole network. He then he identified kind of the choke points of this network of which enzymes were most important at different temperatures. And so he decided to supplant those pathways. And the way they did that is instead of needing the machinery to make a particular metabolite, you just provide the cell with that metabolite in the media, so you're essentially skipping that entire pathway that you think is affected by the temperature. And they showed experimentally that the cells grow faster when they're supplanted, but the cells at 37 degrees had no difference because they didn't need the metabolite because their pathway wasn't affected. So these are kind of all the studies that are in this review. If you want to read more about them in general or in detail, they are available in the review. And there's a lot of work has been done in looking at different components and building these up to small scale modules and building up to large-scale modules in modeling. And systems biology is really just trying to connect all these different approaches, and so you can predict the phenotype or the physiological function. And this kind of goes back to how physics was looking at, taking micro measurements and trying to predict macromolecular properties related to diffusion about 100 years ago. And in biology, we're starting to see that you can use models to take, micromolecular measurements and to make macroscopic observations like physiology. And for metabolism in particular, constraint-based modeling is a method that allows you to do this. So in conclusion, the genotype-phenotype relationship is fundamental to biology, and genome-scale models provide a means for its quantitative description. And what is needed to build, high-dimensional models for this is you need the genome sequence, which is kind of your parts list. You need the polyomic data sets, which are your functional readout of what those different biomolecules are doing. And then you need some sort of modeling framework that is amenable to kind of integrating all of this together and giving you an output. And these case studies, as I showed, allow for genome-scale science when looking at metabolism. And there's some areas that we thought would be very important in the future for this field and in modeling in general. One is just you need more integrative modeling. As you saw, most of the techniques that were done used other types of modeling formalisms. So it's important to become experts in other techniques and to, or at least engage other experts in other techniques to kind of do more integrative modeling. There's going to be expansions in applications of these. Most of these were related to microbes. There's going to be a lot of expansion in human metabolism and human disease, as well as microbiome and community modeling. So it's going to be a very important area going forward. And finally, a lot of the fundamental mathematical assumptions that are in these models will be changed as more data becomes available in the coming years. The first is there's a lot of time course metabolomics data available now, so you can actually skip the steady state assumption and use an unsteady state approach because you know what the metabolite concentration changes are in the cell. And also, as there's more work being done at the sequence level with genomics, the GPRs can be redefined instead of by a Boolean formulation, but actually go to the sequence level and see how that would actually affect. And this is probably will be in tandem with the structural bioinformatics work. Thank you.